Agricultural productivity is persistently threatened by insect pests, which account for significant yield losses globally. Traditional approaches to pest management, such as chemical insecticides, have led to environmental contamination, pesticide resistance, and disruption of beneficial insect populations. Engineering insect-resistant plants with volatile organic compounds, a breakthrough in agricultural biotechnology. The advent of genetic engineering and biotechnology has opened novel pathways for developing insect-resistant crops that mitigate these issues. Among these, the use of volatile organic compounds, VOCs, to engineer plant resistance against insect herbivory represents a revolutionary strategy. VOCs are naturally occurring, low molecular weight organic chemicals emitted by plants that play essential roles in plant defense mechanisms. These compounds serve multiple functions, including direct toxicity to pests, attraction of natural enemies, and priming of plant defenses. Harnessing these natural defenses through genetic engineering can create a new generation of crops with inherent resistance to insect pests, reducing dependency on synthetic pesticides while enhancing agricultural sustainability. Plants have evolved sophisticated mechanisms to counteract herbivorous insects, and volatile organic compounds, or VOCs, are a crucial component of these strategies. When plants are attacked, they emit specific blends of VOCs that serve as chemical signals to deter pests, recruit beneficial predatory insects, or warn neighboring plants of imminent danger. These compounds fall into several major classes, including terpenoids, green leaf volatiles, GLVs, benzenoids, and fatty acid derivatives. A well-documented example is the emission of β-farnesene, an alarm pheromone released by wheat plants when infested by aphids. This VOC not only repels aphids, but also attracts aphid predators such as ladybugs and parasitic wasps, creating a natural biological control system. Similarly, maize plants emit E4, 8-dimethyl-1,3,7-non-atriene, DMNT, in response to caterpillar feeding, which draws parasitoid wasps that attack the herbivore. Such interactions demonstrate that VOCs have evolved to provide indirect plant defenses by enlisting the help of natural enemies. Moreover, some VOCs directly deter insect feeding. Methyl jasmonate, MeJA, for instance, is released by many plant species upon herbivory, and has been shown to reduce feeding behavior in multiple insect species. Studies on tomato plants demonstrate that exposure to exogenous MeJA results in decreased larval growth in the tobacco hornworm, Manduca sexta, due to an upregulation of secondary metabolite production. Studies on tomato plants demonstrate that exposure to exogenous MeJA results in decreased larval growth in the tobacco hornworm, Manduca sexta, due to an upregulation of secondary metabolite production. Advancements in genetic engineering have enabled the manipulation of VOC pathways in plants, allowing scientists to enhance their ability to resist insect attacks. There are two primary approaches to this strategy: overexpression of genes responsible for VOC biosynthesis, and the introduction of novel VOC-producing genes from other species. One of the pioneering studies in this field involved engineering Arabidopsis thaliana to overexpress β-caryophyllene, a sesquiterpene known to attract parasitoids of root herbivores. Native maize lines emit this compound to attract Heterorhabditis megatus, a nematode that preys on corn rootworm larvae. However, many commercial maize cultivars have lost this trait through breeding. Researchers successfully restored this trait by introducing a β-caryophyllene synthase gene, demonstrating that the engineered plants attracted beneficial nematodes and reduced rootworm damage significantly. Another successful case involved cotton plants engineered to emit elevated levels of DMNT and Linalool, both of which have strong repellent effects on cotton bollworms and whiteflies. Another successful case involved cotton plants engineered to emit elevated levels of DMNT and Linalool, both of which have strong repellent effects on cotton bollworms and whiteflies. Field trials revealed that transgenic cotton plants suffered substantially less damage than non-modified plants and had increased populations of predatory wasps. Moreover, rice plants have been engineered to produce β-farnesene, a monoterpene that attracts natural enemies of the brown planthopper, one of the most destructive rice pests in Asia. Studies demonstrated that fields planted with VOC-modified rice had higher predator populations and experienced reduced planthopper infestations compared to conventional rice varieties. One of the most groundbreaking examples comes from tobacco, where researchers introduced a linalool synthase gene from mint, allowing the plant to emit high levels of linalool, a compound toxic to aphids and whiteflies. Greenhouse experiments showed that engineered tobacco plants had significantly lower aphid populations, demonstrating the feasibility of cross-species VOC transfer. While laboratory and greenhouse trials have demonstrated the efficacy of VOC-mediated insect resistance, field trials are essential for validating these approaches in real-world agricultural settings. While laboratory and greenhouse trials have demonstrated the efficacy of VOC-mediated insect resistance, field trials are essential for validating these approaches in real-world agricultural settings. A landmark field trial conducted in Switzerland evaluated maize lines engineered to emit β-caryophyllene. Over 3 growing seasons, researchers observed a 60% reduction in rootworm damage and a 40% increase in beneficial nematode populations compared to conventional maize. Importantly, these benefits were achieved without any adverse effects on non-target organisms, highlighting the ecological sustainability of this approach. Another field study in China focused on transgenic rice plants producing elevated levels of β-farnesene. Over 2 years, farmers using VOC engineered rice observed a 50% reduction in brown planthopper populations and a 30% increase in predator insect numbers, leading to significant reductions in insecticide applications. In the United States, transgenic tomato plants engineered to release DMNT and MeJA were tested in commercial farms, showing a 40% decrease in caterpillar damage while maintaining fruit quality and yield. The engineered tomatoes also had higher levels of defensive secondary metabolites, providing additional resistance to bacterial and fungal pathogens. Such studies confirm that VOC-engineered plants can offer durable and environmentally friendly pest resistance. Despite the promising results, several challenges remain in deploying VOC-based insect-resistant crops on a large scale. One major concern is the potential for unintended ecological consequences. While VOC emissions are generally beneficial in attracting natural predators, excessive or altered VOC profiles could disrupt existing ecological networks, potentially attracting unwanted pests or repelling pollinators. Additionally, metabolic trade-offs must be considered. The production of VOCs requires significant metabolic energy, which could lead to reduced plant growth or yield if not properly balanced. Another challenge involves regulatory approval and consumer acceptance. While VOC-based pest control is more environmentally sustainable than chemical pesticides, concerns over genetic modification persist in many regions. Future research should focus on fine-tuning VOC emissions to maximize pest resistance while minimizing metabolic costs. Advances in synthetic biology could allow for the precise control of VOC release in response to specific pest attacks, reducing the risk of continuous VOC emissions that might have unintended ecological effects. The engineering of insect-resistant plants using volatile organic compounds represents a transformative advancement in agricultural biotechnology. The engineering of insect-resistant plants using volatile organic compounds represents a transformative advancement in agricultural biotechnology. By leveraging plant-derived chemical signaling, scientists have developed a new generation of crops capable of deterring pests, attracting natural predators, and reducing the need for chemical pesticides. Case studies on maize, rice, cotton, tobacco, and tomatoes demonstrate the efficacy of VOC-mediated resistance in both laboratory and field settings, offering promising solutions to global food security challenges. As the world seeks sustainable and environmentally friendly alternatives to traditional pest control methods, VOC-based genetic engineering stands at the forefront of a new era in crop protection, promising safer and more resilient food systems for future generations.